everybody. It's Ashley. Thanks, everyone, for joining us. Thank you again for joining us today. While we can't have our regular lunch and learns, we're lucky enough that we can still do this virtually. And today I have the honor of introducing someone many of you know. Dr. Wisniewski is a board-certified neurologist and neuropathologist. He holds a number of positions here at NYU, including director of our NYU NIH-funded Alzheimer's Disease Research Center, the Pearl Barlow Memory Disorder Center, the New York State Center of Excellence for Alzheimer's Disease, the Conformational Disorders Lab, the Neuropathology Fellowship Program, as well as the director of the Center for Cognitive Neurology. In addition, he's a professor in the departments of neurology, pathology, and psychiatry. Dr. Wisniewski has written over 300 peer-reviewed publications and holds 23 U.S. patents. He's been continuously funded by the NIH for over 25 years, with much of his research focused on developing novel therapeutic methods for Alzheimer's disease. Today, he's going to be presenting on a question frequently asked, not only at our center, but around the world. Will there be a vaccine for Alzheimer's disease? So with that being said, and with great pleasure, I'd like to introduce Dr. Wisniewski.
Hi, everyone. Welcome to this Learn at Home series seminar. I'll be presenting some of our past and present work on this and where the field is moving in this very hopeful time where we're hoping that we'll be developing disease-modifying therapies for Alzheimer's disease, but the challenge is great. So in terms of my objectives for this seminar, they're laid out here. I'll talk a little bit about the basic neuropathology of Alzheimer's disease, what we think are the causes, albeit that's quite complicated, and then also some of the developing vaccine strategies. So in terms of Alzheimer's disease and dementia, there are many causes that, many diseases that can produce cognitive deficits that are a dementia. Alzheimer's disease is certainly the most common of these, but vascular disease, Lewy body disease and Lewy body dementia, frontotemporal dementia, and things like Creutzfeldt-Jakob disease related to prions, all can do it. And there are many things that are shared in the pathogenesis of these causes of these different diseases, but there are also differences. So this is one reason why it's important at the Alzheimer Center and Barlow to get a good diagnostic workup. So these driving forces for ultimately producing a dementia are well defined and established. In terms of the challenge for Alzheimer's disease and the numbers, that's summarized in this slide here. Alzheimer's disease is the 6th commonest cause of death. It's incredibly common in the United States and in the world in general. The costs associated with dealing with Alzheimer's disease are really quite staggering. And the projections by the middle of the century would really bankrupt the healthcare system if we don't produce some therapeutic strategy before that. And these pictures also show what the underlying pathology is, the amyloid plaques, the deposition of amyloid in the blood vessels in the brain, the neurofibrillary tangles, and the inflammation, the gliosis. And all of that produces shrinkage of the brain. When one looks at how medicine in general is doing at treating diseases, at preventing deaths from those diseases, you can see that in many major categories over the last couple of decades, we've done very well at lowering HIV deaths, stroke, and heart disease, and lowering or making inroads in certain cancers. But unfortunately, for Alzheimer's disease, it's the only cause of death among the top 10 for which we really have no effective strategy to treat or indeed to slow it down. So there's a great need really to have some effective therapies here. And that's the challenge. And since we're living in the high stress COVID era, there are also important things to say about the interaction of COVID and dementia and Alzheimer's disease. And patients affected by these disorders are particularly vulnerable to have severe COVID infections. The presentations in patients with Alzheimer's disease are often atypical. So folks that have Alzheimer's disease may present with COVID infection with a change in their mental status. They can become more confused rather than presenting with a fever or a cough. So the diagnosis can be more challenging, at least in the beginning. And also the mortality in this population, in particular in folks who are residing in nursing homes, can be unfortunately extremely high, up to 30 to 40%. So this is an area that we're also working in currently, looking at Alzheimer's biomarkers in patients with COVID infection and seeing how there is an interaction. So this is a topic of great interest and something that we're very actively doing, and it will be the subject of future talks. So the basic pathology in Alzheimer's disease are these plaques. So these brown spots that you're seeing, this is a section of a human brain looked under the microscope, where we've used special stains to outline these accumulations of this toxic protein amyloid beta, and the brain becomes full of these plaques, which are toxic to neurons. So this accumulation leads to the death of neurons. Another major pathology is the accumulation of neurofibrillary tangles, and these are stained neurons. And this brown stuff is the accumulation of tangles in neurons, where the neurons become filled with this abnormally phosphorylated tau protein in the form of neurofibrillary tangles. And again, this is associated with the death of neurons and dementia. Another important pathology in Alzheimer's disease is the accumulation of that amyloid beta protein in blood vessels. So that this circle here is a blood vessel in an Alzheimer's disease patient's brain that's full of amyloid beta, and this amyloid beta is spilling out into the brain parenchyma as well. And this accumulation of amyloid beta can be associated with hemorrhages and strokes in the brain as well. So this is the sorts of pathologies that define Alzheimer's disease. The two culprit toxic proteins that are thought to drive Alzheimer's disease are, again, this amyloid beta protein and this tau protein. Both of these proteins are normal proteins in the human body, but in Alzheimer's disease, they aggregate, they stick together and form these clumps. And these clumps, these small order clumps of amyloid beta and abnormally phosphorylated tau protein, these oligomers, are the most toxic species in Alzheimer's disease, and they're the target of many therapeutic approaches. When these proteins aggregate, clump together in very large aggregates, the amyloid beta forms those plaques that you just saw in the previous slides, or accumulates in the blood vessels in the brain, forming that vessel amyloid. And these very large clumps of the tau protein form the neurofibrillary tangles or the paired helical filaments. This pathology accumulates in the brain over a very long period. So before a patient develops mild cognitive impairment or dementia, the amyloid beta is accumulating in the brain for over 20 years. And then following the amyloid beta, there's also the accumulation of that abnormal phosphorylated tau protein. And that's something that with some of the experimental biomarker studies that we're doing in the Alzheimer's Center and in our clinical trial program, we can diagnose folks with these very early stages of Alzheimer's disease and perhaps start therapeutic approaches at this stage before there is much in the way of cognitive symptoms to help prevent that. And obviously, one wonders in this long buildup of Alzheimer's disease pathology, where certain important political figures are in the spectrum of Alzheimer's disease pathology buildup. And I think that's an open question. In terms of the major forms of Alzheimer's disease, most of Alzheimer's disease is late onset, that is onset after the age of 65, and is sporadic. So it just occurs without a clear family history and without a clear genetic component. There's the less common familial forms of Alzheimer's disease that have an onset at much younger, before the age of 65. And this is linked to mutations in particular genes that are listed here. For the sporadic forms of Alzheimer's disease, there are a number of environmental risk factors that are listed here, but there are also complicated genetic factors that can drive this late onset form of Alzheimer's disease. And these genetic things which drive sporadic Alzheimer's disease are listed here in this kind of complicated slide, but it shows that there are many different genes that have been identified in what's called gene-wide association studies, or GWAS, that can drive sporadic Alzheimer's disease. So the cause of the common forms of Alzheimer's disease have a complicated genetic background. So there's many different things that can lead to those plaques and tangles and vessel amyloid that accumulate ultimately in Alzheimer's disease. The strongest single genetic risk factor for late onset sporadic Alzheimer's disease is the apolipoprotein E gene. And there are 3 different forms of apolipoprotein E in the human population. There's ApoE3, that's the most common. There's ApoE4, that's linked to Alzheimer's disease, and there's ApoE2, which is protective from Alzheimer's disease. And you can see that in comparisons of the general population versus the sporadic Alzheimer's disease population, there's a marked overrepresentation of ApoE4. And you can have 1 or 2 copies of ApoE4. If you have 1 copy, your risk of developing late onset Alzheimer's disease goes up 2 or 3-fold. But if you have 2 copies, it goes up very substantially, 8 to even 12-fold, depending on the population. So this is the amyloid cascade hypothesis, where you have the normal amyloid beta protein, again, a normal protein in the human body that can, in disease states, aggregate to form these oligomers, which are toxic to neurons. and produce neuronal death. They can also make bigger clumps that are deposited in the brain as plaques and accumulation of amyloid beta in blood vessels. And that ultimately drives the accumulation of abnormally phosphorylated tau in neurons and leads to neuronal death. And the presence of ApoE4 promotes this process and tends to promote aggregation of amyloid beta in these toxic species, and also inhibits the clearance of these toxic species from the human brain. So this is the amyloid cascade hypothesis listed here. At the moment, we have those symptomatic drugs that can help somewhat in Alzheimer's disease, like Aricept and Exelon and Namenda, which are very commonly used. But there are also all of these novel therapeutic approaches, in particular immunizations, vaccines, that we and others have been working on for many years. And these vaccine approaches can target with antibodies to these amyloid beta species, and they can target different forms of the amyloid beta. And there have been many experiments in animal models using this approach of targeting amyloid beta. And more recently, there's more approaches that are targeting the abnormally phosphorylated tau and the neurofibrillary tangle approach. In my own lab, we've been developing a very unique approach that potentially targets both of these pathologies at the same time. And that approach is explained in this slide, which is a little bit complicated, but I'll try to explain it to you. We developed this approach by studying patients with a very rare form of dementia called British dementia. And it's called this British dementia because the first cases came from the United Kingdom. And in this rare form of dementia, there's a change in the patient's DNA where a section of the DNA gets translated into a protein from a piece of the DNA that normally is not red. It's a piece of the protein of the DNA that's junk DNA. But in this case, this piece of junk DNA gets converted into a protein that also tends to produce aggregates and oligomers, just like tau and A beta oligomers, but with a different protein sequence. This particular sequence, these letters here, are letters designating amino acids. And these are the building blocks of proteins. So we took this amino acid sequence and formed oligomers made up from this particular small peptide. And these oligomers look exactly like amyloid beta oligomers and tau oligomers, even though the protein sequence here has no similarity to the sequence of amyloid beta and tau. And we use that in mice to do tests with immunization. These were Alzheimer's mice. And we showed that this leads to therapeutic efficacy. So it helps treat the Alzheimer's disease in these model mice. And we've also made antibodies which potentially can be used to treat patients. So this is a novel approach that we've developed. So this approach we used for active immunization. So we gave the peptide as an immunogen, as a vaccine to these Alzheimer's mice. And we checked to see if the mice got smart. And the sort of way we do that is using tests like the radial arm maze, where we put the mouse in the middle here, and there's a reward, a food or a water reward at the end of each of these arms. And the smart mouse can remember which arm they've been down to get the reward. But the dumb Alzheimer's disease mice, they keep on making mistakes and going down the same arm more than once. So the Alzheimer's mice shown in the line in the blue, they make lots of mistakes. They keep on going down the same arm, forgetting that they've been there already. Whereas the treated Alzheimer's mice did much better. And we showed that using a number of different mouse models. We've also used this approach to make monoclonal antibodies. So that those are antibodies that can be given to patients to target these oligomers. And the way we did that is explained in this slide. And it's a complicated slide, but basically what we did is make proteins, peptides, in the shape of amyloid beta oligomers and abnormally phosphorylated tau oligomers, and injected them into mice and induced antibody production. And these antibodies, because they were to this shape that's called a beta pleated sheet structure, it produced antibodies that recognize both those toxic oligomer species of tau and the toxic species of amyloid beta. And this method we published in a number of papers, and also we have a number of patents that are associated with this novel approach that we're taking. And in fact, this listed patent at the bottom here was just issued yesterday. So getting patents allowed from the US Patent Office often takes a while. And this particular patent, we were back and forth with the Patent Office for 3 years, but they allowed it as of yesterday. So, and the method is published there, and it's available for anyone to try out. So we've used some of these monoclonal antibodies to inject into Alzheimer's mice. And this cartoon just depicts one of those experiments where we gave the antibodies that recognize those toxic oligomer species of amyloid beta and tau into Alzheimer's mice and these were quite old mice, 15-month-old mice. That's old for Alzheimer's mice. And at that age, they already have a lot of pathology. What you're seeing here is one of those, a brain section of one of those Alzheimer's mice. And all of that brown stuff is the accumulation of amyloid in the mouse brain. And all of that is toxic to the mouse brain, and it is associated with dementia, cognitive problems in the mice. The ones that we treated with our monoclonal have much less of that brown staining. And you can see the quantitative difference between the untreated mice in the blue and much less of this brown stuff in the treated mice shown in red. And you can obviously see the difference in this histology section, so a section of the brain of this Alzheimer's mice. Another technology that we're developing is something using affibodies. And these are proteins that are kind of like antibodies that the body produces, but they're much smaller in size, many-fold smaller. So these smaller proteins are more able to cross the blood-brain barrier, to get into the brain. And also these types of antibody-like antibody mimetics are already used in humans for a variety of approaches like chemotherapies in cancers, as well as in imaging. So these sorts of affibody approaches are already safe and used in people. And we designed a specific antibody, which is shown in this cartoon, that recognizes the amyloid beta protein, which is one of the proteins that accumulates in Alzheimer's disease and causes problems. And we used it to treat these Alzheimer's mice here. That's someone in my lab getting ready with the antibodies to inject in these mice. And again, in this radial maze for the mice, the treated animals made far fewer errors. So the Alzheimer's mice got smarter with this sort of treatment. And we're now developing a number of different antibodies that can recognize different toxic proteins in the human brain, with the potential of using this approach for Parkinson's disease, where the target is alpha-synuclein, where alpha-synuclein accumulates in Parkinson's disease, and also to another protein, TDP43, that accumulates in frontotemporal dementias. So this is a very versatile tool to make vaccines for different causes of dementia. Another approach that's a form of a vaccine that we're working on is to stimulate the innate immune system. And every animal that exists has some type of innate immune system. The innate immune system is something that's always ready in our bodies to fight infection. It's the first line of defense. It works through receptors that recognize pathogens, so things like bacteria and viruses, and helps fight those off. And in particular, macrophages are an important part of the innate immune system, and they circulate in our bodies and act to immediately fight infection as part of the innate immune system. One way of stimulating the innate immune system is with using CpG oligonucleotides. And all that means is that they're small pieces of DNA that are like DNA from bacteria. So when the body sees these little pieces of DNA, it's like being invaded by a bacteria, and that stimulates the macrophages to be hyperactive and better at clearing these pathogens. And these CpG oligonucleotides are already used to harness the innate immune system in cancer therapies and in a number of human vaccines and in a variety of autoimmune problems. So we thought that it might be of value to try using this approach in Alzheimer's disease. So we used these CpG, these bits of DNA, and we injected them into different Alzheimer's disease models. One Alzheimer's model is called TG2576. And when we gave the CpG, it reduced those plaques. So on the left is a control animal, and you can see a lot of those plaques, and then treated as fewer. And this is a different type of Alzheimer's model where there's both plaques and the tau, those tangles. And the CpG led to reductions in both of those pathologies, as shown here. And more recently, we've turned to a natural model of Alzheimer's disease, aged squirrel monkeys. And these, obviously, monkeys are a lot closer to people than mice are. So it's important to test some of these treatments in animals that are more like people, because if they work well in these animal models, then they're more likely to translate effectively to people ultimately. And squirrel monkeys, shown here, they develop some Alzheimer's related pathology. And these are pictures of amyloid accumulating in old squirrel monkeys. And this looks just like what happens in the human brain. So this accumulation of amyloid in the blood vessels in the monkey brain looks identical to what happens in people. So it's a good model. And this produces a dementia in the old monkeys. So here you're seeing we're testing these monkeys in a cognitive test where underneath this bucket there was a grape reward. And these monkeys are quite fond of grapes. There was one side on this bucket, inverted bucket, where the monkey could get the grape. And in this cognitive test, we would switch the side where the opening is. And the Alzheimer's old monkeys were really bad. They made lots of errors in working out when we switched the side. They couldn't figure it out. They made lots of errors. Whereas the treated monkeys did much better. And this graph here depicts that the old Alzheimer's monkeys performed like they were rejuvenated. They performance on this task was like a young monkey. And the other test that we've developed, these monkeys like to do finger painting. So the younger monkeys and the smarter monkeys can make more complicated finger drawings like here, whereas the Alzheimer's monkeys made rather simpler drawings. So this is another test that the monkeys enjoyed doing these sorts of tests more than the bucket test. So there was all sorts of ways of quantitating cognitive benefits. But ultimately, we looked at the brains of the monkeys, and in the monkeys that received the treatment, the CPG, there was a lot less of the plaques, this black stuff that you're seeing on the left in comparison to the controls. So we think that this is a novel approach that's been highly effective in the monkeys, and we will be starting a phase 1 clinical trial of this approach at our Alzheimer's Center, hopefully later this year, since we've shown that this is highly effective in various mouse models and also in the monkeys. Overall, there are many, many different clinical trials that are being done right now across the United States. And this slide depicts the various clinical trials, and there are many different targets that are being studied in these clinical trials sponsored by the National Institutes of Health, specifically the National Institute of Aging. And we're involved in many of these in our Alzheimer's Disease Center. So this is a hopeful time where many different approaches are being tried. And in our own clinical trial program, we firstly evaluate patients in Barlow, work out an accurate diagnosis, and then make referrals to with folks who are interested in being part of these clinical trials, and make sure that the subject is appropriate for any given trial. In these coming slides, I'm just summarizing some of the clinical trials that we're enrolling patients in right now. So we have an intervention of exercise to see if that can help with reducing Alzheimer's related pathology. We're also doing various imaging studies to just observe how various biomarkers develop over time as the disease progresses. We're studying an antiviral agent, valacyclovir, to see if that can be effective in Alzheimer's disease, as well as looking at the effects of giving nicotine patches, which can reduce the production of amyloid beta. And then we have a number of monoclonal antibodies. So these are injections of antibodies, in this case, to amyloid beta, to prevent and clear those amyloid plaques. And again, we're recruiting patients for those sorts of clinical trials. The antibody that's garnered a lot of interest right now is an antibody called aducanumab, and the company involved here is Biogen. And what they did here is look at healthy, aged individuals to see if they had any naturally occurring antibodies that recognize those abnormal oligomeric clumps of amyloid beta, thinking that in folks that already naturally produce those antibodies, that could be a form of being protected against developing Alzheimer's disease. And they found that indeed some elderly individuals had such antibodies, and through a genetic means, those antibodies were cloned, and monoclonal antibodies were produced and then tested in Alzheimer's disease mice, and ultimately developed in a form that could be used as a treatment. So this particular aducanumab, when it was made into a mouse monoclonal, the mice were injected with this antibody, it was shown that the antibodies latch on to the amyloid plaque here, this accumulation of amyloid beta, and that antibody sticking on to the amyloid beta produced stimulation of macrophages in the brain, which are the microglia, and that induced those cells to gobble up, to eat up the amyloid plaques, which is what you're seeing here. These macrophages are sticking on to the amyloid plaque and clearing it through eating it up, through digesting it. So there was a phase 3 clinical trial of this antibody, aducanumab, and the scheme of giving the antibody at different doses, at 3, 6, and ultimately 10 milligrams per kilogram, is depicted here. And this is a clinical trial that ran over 3 years. And the results on the cognitive testing, and one of the cognitive tests is listed here, CDR, sum of boxes. In the patients that got treated, there was a marked cognitive benefit. And this is just showing the statistics of it, that this was highly statistically significant, with a 23% difference between the control patients and the patients that received the treatment. So this is something that's likely of clinical significance. When we looked at, in this trial, when it was looked at the amyloid burden in the brains of the patients using PET, the treated patients had a marked reduction of the amyloid beta as shown in this graph. And that was dependent on the dose of the antibody. So the folks that got the highest dose, 10 milligrams per kilogram, had the greatest reductions of amyloid beta. And the folks that got a lower dose of the antibody also had a reduction, but it wasn't as big. And importantly, there was also reductions of the abnormally phosphorylated tau protein, which is the other pathological protein that accumulates in Alzheimer's disease. And that was also reduced in a dose-dependent fashion. So these are results that are highly supportive of this antibody perhaps being a treatment for Alzheimer's disease. So this slide shows the conclusions that really this is showing a lot of promise. We're running another phase 3 clinical trial where we're redosing folks that previously were part of the aducanumab clinical trial and giving them the opportunity to receive that antibody as part of a continued clinical trial. But earlier this month, Biogen applied to the FDA for approval for aducanumab as a treatment for Alzheimer's disease. And that's the first time there's been an application like this for a new agent to potentially treat Alzheimer's disease that's a biologic since 2003. So it's possible that the FDA will, in fact, approve aducanumab as a potential disease-modifying treatment for Alzheimer's disease, although that's a process that often takes quite some time, a year or two. So this is the contact information for our clinical trial program, but also there's lots of very good information about clinical trials that are available throughout the United States and in the tri-state area. And these are websites where that information is available either through the Alzheimer's Association or through the National Institutes of Health, the Institute on Aging, and also clinicaltrials.gov. And so I would say in conclusion that we're in a hopeful time. There's a great need for effective therapies for Alzheimer's disease. And these immunotherapeutic approaches, these various vaccines that we're working on in the lab and looking at in various clinical trials are now beginning to show promise. And we're very much hoping that perhaps that we're beginning to see the light at the end of the tunnel. But unfortunately, there's still a ways to go. These things take a lot longer to come to fruition than one would like. But that's the reality. And also, it reflects the complexity of the problem that Alzheimer's disease is. So this just highlights some of the Alzheimer's Research Center team in happier days from last year where social distancing wasn't a requirement. So it's going to be a long time before we can take another picture like that, unfortunately. So happy days. But certainly these are interesting times we live in. And in terms of a vaccine for Alzheimer's disease, certainly very hopeful, although we have yet to see if this will really come to fruition. So I'm happy to take questions now.
Yeah.